morning, everyone. So already we have discussed all important short answer questions and long answer questions also we have discussed in the previous sessions. So today, let us go with important VSQs, that is very short answer questions. And in today's sessions, I would like to discuss all the questions, 10 questions that which have been given in your recent IPE exam, whatever happened, 2023 IPE exam, whatever questions they have given VSQs. Today we will discuss them because there is chance that at least some 2 to 3 VSQs again they may ask. So same questions again they may ask, at least expecting minimum 2 and maximum 3 questions, or if time is good, even more questions also may come, same questions. So let us discuss the important VSQs that which have been given in the recent IPE exam, whatever happened. Okay, so that is 2023 question paper. So in that first question, already you would be knowing dental formula of human beings. So give the dental formula of adult human. So when they ask this VSQ, what you will write? First of all, you should write what is meant by dental formula of adult human. So dental formula means what? Arrangement of teeth. So that is arrangement of teeth in each half of the arrangement of teeth in each half of the upper and lower jaw. Upper and lower jaw of adult human beings. Upper and lower jaw of adult human beings is only called as dental formula. So first of all, definition you should write. Arrangement of teeth in each half of the upper and lower jaw of adult human beings is only called as dental formula. That means this is our upper jaw and this is our lower jaw. So in the upper jaw, if you take only half of the upper jaw, so how the teeth are arranged and what are, how many are arranged and what are they? So the arrangement of teeth in each half of the upper jaw and lower jaw is only called as dental formula. So that to know, in the arrangement of what? In the order of incisors, canines, premolars, and molars. So this should be the arrangement. So actually the arrangement is incisors, canines, premolars, and then molars. And the dental formula is 2, 1, 2, 3 by 2, 1, 2, 3. So that means here incisors in each half of the upper jaw 2, in each half of the lower jaw 2, canines in each half of the upper jaw 1, in each half of the lower jaw 1. The next premolars in each half of the upper jaw and in each half of the lower jaw premolars, and molars in each half of the upper jaw and in each half of the lower jaw. So 3 by 3. In that, no, last molar teeth or third molar teeth is only called as wisdom teeth. So third molar teeth is called as wisdom teeth. So this wisdom teeth, generally it erupts after the age of 17. So after 17 years only it starts to erupt. It may erupt at any age after 17. Like it may be at 20 years or 25 or 30 or 40. It may erupt at any age. So like this, the dental formula is 2, 1, 2, 3 by 2, 1, 2, 3 of adult human beings. So where I is equal to what? I is equal to incisors and C is equal to canines, and then PM is equal to premolars and M is equal to molars. So this is the first question answer. So give the dental formula of adult human means the arrangement of teeth in each half of the upper and lower jaw of adult human beings is only called as dental formula. That too in the order of incisors, canine, premolar, and molar, which is 2, 1, 2, 3 by 2, 1, 2, 3, where I is equal to 2 by 2, C is equal to 1 by 1, PM, premolar is equal to 2 by 2, and again M, molar is equal to 3 by 3. So that means here I stands for incisors, C stands for canines, and PM stands for premolar, and M stands for molar. So when they give this question, you are supposed to write. So like this, the answer. Hope that you have understood this. Now let us go with, so the next question. What is the next question which have been given in the question paper? Define glomerular filtration. So next question is glomerular filtration. The definition you should give. That is
Hello. Hello. Hello, ma'am. Hello. Ma'am, I can't hear your voice.
Oh, they have raised their hand. Am I audible now, children? Am I audible now?
Yes.
Yes?
Yes.
Am I audible now?
Yes, ma'am.
Okay, ma'am. Okay, thank you. And what about the board clarity? Is it clear?
No.
Board clarity is not there. Board clarity is not there, they are saying. And even a little more, I should write this side, I think so, because see, that has got cut. You just sit and check here, no? Like how it is coming. Okay, fine. So differences between actin and myosin. Let me write this side again because it is getting cut, no? That side. So little, let me write this side. So third question we are discussing, third VSAQ, which has, which they have given in the board exam. Differences between actin and myosin. So actin is a thin filament. Actin is a thin filament. And whereas myosin is a thick filament. This is the first difference which you should write. Actin is a thin filament, whereas myosin is a thick filament. Okay, thin filament and then thick filament. Then next question, second point. Actin, it is a protein which is present in light bands. Which is present in light band. And whereas myosin is a protein. Myosin is a protein present in dark band. Present in dark band. Here you may get a doubt like what is this light band and dark band. So actually actin and myosin are the proteins which are present in skeletal muscle. They are called as contractile proteins. That means these proteins are only responsible for muscle contraction. So when we take a skeletal muscle, which is long, which consists of muscle fibers, long cylindrical muscle fibers, okay, each muscle fiber, again, it contains alternate dark and light bands. For example, if it is dark colored band, myosin. For example, if this is dark colored band myosin, it contains again light color band alternately like this. And again, what happens? Again, dark colored band. Then again, light colored band like this. So that means both dark and light colored bands, they are present in alternate fashion. So in the dark colored band, which is called as, the myosin protein is present. In the dark colored band only, the myosin protein is present. Whereas in this light colored band, the actin protein is present. Actin protein is present. So then next coming to, here in case of actin, the filaments are attached to Z line. Filaments are attached to Z line. Already in the structure of the skeletal muscle tissue, you would have learned that. So for example, if you take this thin filaments, actin filaments, so here at the center, they contain a fiber which is called as what? Z line. So it contains what? Z line. So like this. On this side also, this is also actin filament where at the center it contains Z line. So that means the actin filaments are connected to Z line. And whereas in case of myosin, the myosin filaments are connected to M line, membranous line. So myosin filaments are connected to M line. So M line means what? Membranous line. So like this, three differences you are supposed to write. What are they? Actin is a thin filament, whereas myosin is a thick filament. Then actin is a protein present in light colored band, whereas myosin is a protein present in dark colored band. Then actin filaments are attached to Z line, and whereas myosin filaments are connected to M line. Understood? So these are the differences between actin and myosin. So first question we have learned about dental formula, which is 2123 by 2123 in case of adult human beings, the dental formula of permanent teeth. And second question we have learned about what is glomerular filtration, the definition for that, and glomerular filtration rate is 125 ml per minute. So that is equal to 180 liters per day. And third question, differences between actin and myosin. So hope that it's clear for you. The next coming to fourth question. So fourth question is, name the keystone bone of the cranium and where it is located. So already you know that in the cranium, bones are present. So like sphenoid bone, ethmoid bone, parietal bone, temporal bones, occipital bones, frontal bones. So totally 8 cranial bones. Totally in cranium, how many bones are present? 8 bones are present. In that, which is called as keystone bone of the cranium. So in that, which is called as keystone bone of the cranium. So keystone bone of the cranium is sphenoid bone. So let me write the question. Which is called as keystone bone of the cranium. Which is called as keystone bone of the cranium. And where is it located? Which is called as keystone bone of the cranium. And where is it located? So what is the answer which you should write for this? Sphenoid bone. Sphenoid bone is called as keystone bone of the cranium. Sphenoid bone is called as keystone bone of the cranium, and they have given where it is located. It is located at the base of the skull. Where it is located? It is located at the base of the skull. It is located at the base of the skull. So, like this, you are supposed to write keystone bone of the cranium is sphenoid bone. It is located at the base of the skull. That's all. If you write 2 points, 2 marks you will get. 1 mark for sphenoid bone and 1 mark for location, which is located at the base of the skull. So it is located at the base of the skull, sphenoid bone. The next coming to another question. Fifth question. Write the names of any 4 mononuclear phagocytes. Write the names of any 4 mononuclear phagocytes. So what are 4 mononuclear phagocytes? So totally 4 phagocyte names you should write. Mononuclear phagocytes. Actually many are there. You can write any 4. So fifth question. Names of any 4 mononuclear phagocytes. So in that, first you can write Kupffer cells present in liver. So Kupffer cells in liver, present in liver. The next, monocytes present in bone marrow. Monocytes present in bone marrow. Then histiocytes present in connective tissue. Histiocytes which are present in connective tissue, and macrophages which are present in adipose tissue. Adipose macrophages which are present in adipose tissue. Like that, you can write the names of 4 mononuclear phagocytes. So, it is Kupffer cells present in liver, monocytes in bone marrow, histiocytes in connective tissue, and then macrophages in adipose tissue. So, these are 4 mononuclear phagocytes. So, for each name, half mark. If you write 4, then only you will get 2 marks. So, correctly, if you write, then you will get 2 marks. So, fifth question, names of any 4 mononuclear phagocytes, Kupffer cells in liver, monocytes in bone marrow, histiocytes in connective tissue, and then macrophages present in adipose tissue. So, the name itself indicates what is meant by phagocyte. Phago, eating, cyte, cell. When any foreign agents like pathogens or germs enter into our body, so then they will be non-specific immune cells. These are where they will be eating away them, okay, which are present in different tissues in different forms and names, like Kupffer cells in liver, monocytes in bone marrow, then histiocytes in connective tissue, and then macrophages in adipose tissue. The next, coming to another question, another important question. So that is 6th question. What is erythropoietin and what is the function of erythropoietin? So next question is erythropoietin and its function. So erythropoietin is a hormone. So actually this question they have given from endocrine system chapter. Endocrine system chapter. So 6th question. What is erythropoietin? And mention its function. Erythropoietin, mention its function. So what is erythropoietin? Erythropoietin is a hormone. Erythropoietin is a hormone which is secreted by JG cells of kidney. So what is JG cells? Juxtaglomerular cells. So juxtaglomerular cells. So hope that you can't see this side. So let me write here. So they are present in JG cells of the kidney. They are secreted by juxtaglomerular cells of kidney. Juxtaglomerular cells of kidney. And the function of this erythropoietin, the name itself indicates erythro, red blood cells, poiesis, production. So erythropoietin, it is mainly involved in proliferation and differentiation. Proliferation and differentiation of erythroid progenitor cells. That means precursors. Erythroid progenitor. From this only RBC will be formed. So for the maturation and differentiation of what? Erythroid progenitor cells. So erythropoietin is a hormone which is secreted by juxtaglomerular cells of the kidney, and the function is what? Proliferation and differentiation of erythroid progenitor cells. So that is erythropoietin. Hope that you are writing. Juxtaglomerular cells of the kidney, then proliferation and differentiation of erythroid progenitor cells. So each point carry one mark. So each point carries 1 mark. So totally 2 points. For secretion, one mark, and for the function 1 mark. So totally you will get 2 marks for this question. Okay. The next, coming to 7th question. The next question. Next question is define spermiogenesis and spermiation. So you should write what is spermiogenesis. Define spermiogenesis and spermiation. So what is spermiogenesis? Spermiogenesis. See, actually, most of the technical terms only they will make us to understand the meaning or the definition of it, right? So spermiogenesis. The transformation of spermatids. The process of transformation of spermatids into the process of formation of spermatids into sperms or spermatozoa. So spermatozoa or sperms is only called as spermiogenesis, the process of formation of spermatids into sperms. So that means from the spermatids, whatever the sperms are getting formed, so that is only called as what? Spermiogenesis. So during the process of spermatogenesis process, first the sperms are formed in the form of spermatids. Now the transformation of this spermatids into sperms is only called as spermiogenesis. The process of transformation of spermatids into sperms is called as spermiogenesis. The next, spermiation. What is meant by spermiation? Once the sperms are formed, so the process of release of sperms, from the seminiferous tubules. Actually, you know that sperm formation takes place in seminiferous tubules, right? So the process of release of sperms from the seminiferous tubules into Sertoli cells. You know that Sertoli cells is another type of cells which are present in the seminiferous tubules. So now the process of release of sperms from the seminiferous tubules into Sertoli cells. From the seminiferous tubules into Sertoli cells is only called as spermiation. So spermiogenesis is the process of transformation of spermatids into sperms, and spermiation is the process of release of the sperms out of the seminiferous tubules. Actually, no, the heads of the, for example, if this is Sertoli cell, the head of the Sertoli cells, actually they are attached to this. The heads of the sperms are attached to this. Now, from the Sertoli cells, they will get detached, and finally, they are moving out of the seminiferous tubules. So that is only called as spermiation. So you should write the definitions for spermiogenesis and spermiation. Spermiogenesis, the process of transformation of spermatids into sperms, and spermiation, the process of release of this out of the seminiferous tubules. The process of release of sperms out of the seminiferous tubules is only called as spermiation. The next question, define amniocentesis. Define amniocentesis and what are the disorders, that which can be detected by this technique. So 8th question, define amniocentesis and name disorders. Name the disorders which can be identified by this technique. Name the disorders which can be identified by this technique. So, amniocentesis, amnion. Generally, where this amnion layer will be present? Amnion layer, it is present surrounding the growing embryo. Already you know 4 extra embryonic membranes like chorion, amnion, allantoin, yolk sac in case of the mammals, right? So, amniocentesis is a technique which is used to detect the genetic disorders present in the growing fetus. It is a technique which is used to detect. Amniocentesis is a technique which is used to detect the genetic disorders or the genetic defects present in growing embryo, or fetus is only called as amniocentesis. Actually, with this technique, so they will be collecting the specific cells which are present in the amniotic fluid, the stem cells, and in that stem cells mainly they will detect the genetic defects. So that means the abnormal genes they will be identifying, so that they are able to prevent many genetic disorders at the embryonic condition, fetal condition itself. So what are the examples? Down syndrome, all these are genetic defects, right? Chromosomal disorders. Down syndrome, Edwards syndrome, and Turner syndrome. Down syndrome, Edwards syndrome, Turner syndrome. So these are some of the genetic defects present generally in humans from the before the birth itself. So like this, amniocentesis is a technique which is used to detect the genetic disorders present in the growing embryo or fetus. Example is Down syndrome, Edwards syndrome, and then Turner syndrome. So for definition, 1 mark, and for examples, whatever you are giving, correct examples, for that 1 mark. So totally 2 marks. Amniocentesis, mainly used to detect the genetic disorders. With examples, Edwards syndrome, Turner syndrome, Down syndrome, Klinefelter syndrome, like this many examples we can give. The next question, define apiculture. 9th question, which had been given in your question paper. Define apiculture. So what is apiculture? Apiculture. So 9th question, define apiculture. Apiculture is rearing of. Apiculture is rearing and maintenance of honeybees and beehives. Honeybees and beehive. Beehive means what? The place where the honeybees are grown. It is only called as beehive. So it is the process of rearing and maintenance of honeybees and beehives. For what purpose? For the production of commercial products, that which we are getting from honeybees. For the production of commercial products. Already in SAQ also we have learned economic importance of honeybees. There I told you different commercial products which we get from honeybees. Like honey we will get, bee wax we get, propolis we get. And you know that honeybees acts as main pollinators. And even the honeybee venom is used in the treatment of arthritis, joint pains also I told you, right? So like this, apiculture is the process of rearing and maintenance of honeybees and beehives for the production of commercial products like honey, bee wax, etc. So such kind of commercial products we will get from honeybees. So commercial means what? We can earn Actually, by selling such kind of products, we can make profits, we can earn money. So that is the reason why they are called as commercial products. So like this, apiculture is the rearing and maintenance of honeybees and beehives for the production of commercial products like honey and then bee wax. So this is only called as what? Apiculture. So for this definition, you will get 2 marks if you write like this correctly. So the next coming to last question, 10th question. What does ADA stand for? And deficiency of ADA causes which disease? Next question. What does ADA? Adenosine deaminase. What does ADA stands for? And deficiency of ADA will lead to which disease? So that is the question. So 10th question. What does ADA stands for? And deficiency of ADA, deficiency of ADA leads to which disease? So this is the question. So first of all, you should write what is meant by ADA. That is adenosine deaminase. Adenosine deaminase. Adenosine deaminase. So ADA stands for adenosine deaminase. Actually, this enzyme deaminase, this enzyme is mainly required for the process of maturation and differentiation of lymphocytes. So during the process of maturation and differentiation of this lymphocytes, ADA enzyme is required. Adenosine deaminase enzyme is required. But the deficiency of this ADA will lead to a disease. Deficiency of ADA leads to severe combined immunodeficiency. In shortcut, we can call it as what? SCID. Severe combined immunodeficiency. Severe combined immunodeficiency. The name itself indicates, right? So severely, it is associated with immune deficiency. Combined means what? Not only a specific lymphocyte. So all the lymphocytes like B lymphocytes and different types of T lymphocytes, everything they will become short in the immune system so that our immunity will get lowered. You know that whenever our immunity get lowered, what happens? We will become opportunistic vehicles. Our body will be something like welcoming now all the antigens, all the foreign agents into our body. So that's why it is called as what? Severe combined immunodeficiency. Such kind of people mainly, they will be having very lower levels of immunity so that they will get any kind of infection very easily. So that is the meaning. ADA is adenosine deaminase, and the deficiency of this ADA, it results in what? SCID, that is severe combined immunodeficiency. So that what happens? The lymphocytes will not undergo maturation and differentiation. And whenever specific lymphocytes are not there, so what happens? The immunity will become lowered so that easily the people will be getting diseases. So any kind of infection. So deficiency of ADA leads to SCID, severe combined immunodeficiency. So for ADA 1 mark and for SCID 1 mark. Totally 2 marks you will get when you write this answer. So these are the 10 questions which they have given in your recent question paper, VSAQs. So definitely you should learn all these 10 VSAQs so that minimum 2, maximum 3 to 4 questions you are having chance to get back again. So that's why don't neglect, learn all these 10 questions. So just we will revise whatever we have discussed today, all the 10 questions. So in that, what is the first question? First question is dental formula of adult human beings. So you should write the definition. So what is definition? That is the arrangement of teeth in each half of the upper and lower jaw of adult human being in the order of incisor, canine, premolar, and molar is only called as dental formula, which is 2123 by 2123. So that is the first question. And next, second question, what you have learned? Differences between actin and myosin. So actin is a thin protein, myosin is a thick protein. Actin is present in light colored bands, whereas myosin is present in dark colored bands. And in actin, the filaments are connected to Z line, and whereas in case of myosin, the filaments are connected to M line. So this question you have learned. And next question is glomerular filtration. So what is glomerular filtration? The amount of filtrate formed by both the kidneys per minute is only called as glomerular filtration, which is equal to 125 ml per minute. So that is 180 liter per day. So that question you have learned. And after that, you have learned about erythropoietin. So what is erythropoietin? Erythropoietin is a hormone which is secreted from JG cells, juxtaglomerular cells of the kidney. It is mainly required for proliferation and differentiation of erythroid progenitor cells, from where this RBC can be produced. So that question you have learned from endocrine system chapter. And after that, even you have learned other questions like name the keystone bone of the cranium and where is it located. So sphenoid bone is the keystone bone of the cranium. It is located at the base of the skull. So that is the question. And next, amniocentesis. What is amniocentesis? It is a technique which is used to detect the genetic disorders in the growing embryo. So what are the different disorders? Like Edwards syndrome, Down syndrome, Turner syndrome. All these are the different genetic disorders. Then what is ADA? What does ADA stand for? ADA is adenosine deaminase enzyme, and deficiency of this enzyme will lead to a disease called as severe combined immunodeficiency, so that the people will have lowered immunity where they will become susceptible for the infections. And next, name any 4 mononuclear phagocytes. So you can write any 4 phagocyte names which are present in different tissues, like histiocytes in connective tissue, Kupffer cells in liver, macrophages in adipose tissue, and monocytes in bone marrow. So like that, you can write the names of 4 mononuclear phagocytes. And after that, other questions which you have learned are differences between spermiation and spermiogenesis. So what is spermiogenesis? The process of transformation of spermatids into sperms is spermiogenesis. And the process of release of the sperms from the heads of the Sertoli cells out of the seminiferous tubules is called as spermiation. So like that, totally 10 questions you have learned in today's session. In tomorrow's session, chapter wise, let us learn other important VSAQs. Okay, so that easily you can score marks in VSAQs, very short answer questions. Now I want, if you have any doubts, kindly ask. Raise your hand so that we will unmute. Any doubt? Siddhika. Only she is there today. Any doubts, Siddhika?
Ma'am, you can revise LAQ questions.
LAQ questions you want revision?
Yes, ma'am.
Okay. So in tomorrow's session, let us do one thing, like not all LAQs at once. So tomorrow one LAQ I will revise along with some very short answer questions. Like that we will finish it off. Each day one LAQ. Okay. Totally 4 are there, no? So each day one LAQ. Is that fine?
Yes, ma'am.
Because we need to cover even very short answer questions, no? That's why.
Okay, ma'am.
Okay, fine. Any other? Okay. Fine, children. Thank you then.